And here we are, back with lecture 14. This is really strange not getting to see everyone, and I'm just now kind of getting used to it, and I hope that you guys are too. So before working through this lecture, and in particular before working through the first part of this lecture, which has to do with meiosis, you should have watched the two meiosis videos. I do hope you enjoyed them. I hope you especially enjoyed the Amoeba Sisters in the second video. And don't let how cute those Amoeba Sisters are fool you. There is a lot of good information here, and it's very well presented. And I would say that's true actually of both videos, even if the first one isn't quite as cute. I suggest that after you've worked through this meiosis presentation, you go back and watch the videos again just to reinforce things. Let's jump in then to 22.1, meiosis and sex cell production. And if you think about it, those two terms pretty much mean the same thing because meiosis is the process of producing sex cells. Male sex cells, of course, are called sperm cells, and female sex cells are egg cells. And then notice the other term for egg cells is oocytes, and the literal translation here is egg cells. Following along, sex cells are produced by a special type of cell division called meiosis and have only one set of genetic instructions carried on 23 chromosomes compared to two sets on 46 chromosomes for all other body cells. And we then use the term diploid for a cell which has 46 chromosomes, and we use the term haploid for a cell which has only 23 chromosomes. This means then that the only haploid cells in your body will be egg cells or sperm cells, and all other cells of your body will be diploid. Moving on then, meiosis 1 separates pairs of chromosomes called homologous chromosomes, and you may recall this term from one of the videos. And think about the homo meaning the same, or at least very similar. So these homologous chromosomes, their members are similar because each pair has essentially the same genes, and on each of these homologous chromosomes, the genes are found at the same location along that chromosome. But these homologous chromosomes are not necessarily identical, and actually won't be identical in any case, because a gene may have different forms, may have variant forms called alleles. So alleles, different forms, alternate forms of the same gene. And back to the outline then, at fertilization, a sperm cell may contain one allele for a particular gene, while an egg cell may contain a different allele. Of these 23 pairs of chromosomes then in a diploid cell, 22 pairs are fully homologous, meaning they contain the same genes, again in the same positions, on a pair of chromosomes, on that pair of homologous chromosomes, while one pair, the sex chromosomes, may not be fully homologous. And the reason for this is that the Y chromosome is much longer than the X chromosome. And if you think about it, that additional length on the Y chromosome means there is additional room for there's additional room for genes on that Y chromosome, and there wouldn't be room for all of these additional genes on that shorter X chromosome. So talking now about meiosis, and remember from the videos that meiosis occurs in two different phases, and these phases are meiosis 1 and meiosis 2. Each cell then undergoes meiosis 1, producing 2 cells, each of which is haploid, so it has 23 chromosomes. Each of these 2 cells being produced by meiosis 1 are haploid, and each chromosome consists of 2 identical DNA molecules, and notice in parentheses some interspersed proteins. Each half of a chromosome then, so essentially each DNA molecule, if we disregard the proteins, and we will because we're just talking about the DNA here, so each half of a chromosome, essentially each DNA molecule, is called a chromatid, and the 2 identical chromatids that make up a chromosome are called sister chromatids. Again, a lot of this should be ringing bells from the videos, and do go back to the videos after you've worked through this lecture. Talking now about the 2 phases of meiosis, the 2 stages of meiosis, if you will, so meiosis 1 and meiosis 2. And notice here now, for the phases of meiosis 1, you're being directed to page 826 and figure 22.1. And if you take a look now at that figure, notice then we're beginning this process of meiosis with a diploid cell, way off to the left, this cell with 46 chromosomes. And notice each of these chromosomes contains 2 chromatids, contains 2 sister chromatids. So each of those X-shaped structures is a chromosome, and again, each half of the X is a sister chromatid. And again, recall these 2 sister chromatids making up any one of those X-shaped chromosomes. These 2 sister chromatids are essentially identical DNA molecules. That's in your notes. We just went over that, so don't worry too much about jotting it down again. But again, with each of these 46 chromosomes that we're starting with, each consists of 2 identical sister chromatids. If we follow the arrow now from the cell on the left to the next cell being shown, and notice up above it's saying first meiotic division, so this is meant to represent meiosis 1. This second cell in line is meant to represent meiosis 1. And notice in these diagrams, or at least in this particular diagram, they're not going through all the different stages, all the different phases of meiosis 1. So first meiotic division, again, just implies that this is meiosis 1 occurring. And then notice now, off to the right, we now have, at the end of meiosis 1, 2 cells, each of which is haploid with 23 chromosomes, each chromosome containing 2 chromatids. We're halfway through meiosis, and so now each of these cells formed by meiosis 1 undergoes meiosis 2, and meiosis 2 then, and let's not worry about the handout for just a minute, let's not worry about the lecture outline for just a minute, but notice now that each of these cells, which has been produced by meiosis 1, by the first meiotic division, each of these 2 cells now undergoes meiosis 2, and this is where you're seeing second meiotic division as the label. And again, they're not showing prophase 2, metaphase 2, anaphase 2, telophase 2, but these are all kind of to be mentally combined, I guess, without a lot of detail by you guys. But again, second meiotic division, we're just inferring now that here meiosis 2 has occurred. And notice then, at the end of meiosis 2, we now have, from that single cell that we started with, we've gone from 46 chromosomes to 23 chromosomes. So we've gone from 1 diploid cell to 4 haploid cells. And notice also now, back to those 4 cells that we end up with, in each of those cells, we have 23 chromosomes, and each chromatid is now considered to be an independent chromosome. So it's a little bit strange, but again, at one point, those chromatids were joined together as sister chromatids to form what we call a chromosome, and now at the end of the process, each individual sister chromatid, once it's separated from its sister, once it's gone to a different cell than its sister, each of what we did call sister chromatids is now referred to as a chromosome. Under phases of meiosis 1, back to your handout, prophase 1, and there's a note here that synapsis and crossing over both occur during prophase 1. If you take a look on pages 826 and 827, and take a look then at figures 22.2 and 22.3, you'll see synapsis being diagrammed. You'll see crossing over being diagrammed. And synapsis then implies, remember synapse, 2 things coming together. So synapsis implies 2 homologous chromosomes are coming together. And when we look then at figure 22.2, notice we're showing a pair of homologous chromosomes that have synapsed, that have undergone synapsis. And as we follow those chromosomes then from A to B to C in the process of crossing over, you can see, and again recall from your meiosis videos, that crossing over involves 2 homologous chromosomes trading pieces, and in the process of trading pieces, they are trading genes. There's a train coming by. I'm going to cut it off here for lecture 14.